Okay, think about this for a second. How does life build its most complicated machines? I mean, there's no factory, no assembly line. It just happens spontaneously inside every single one of your cells. Today, we are diving into the absolutely incredible world of proteins and tackling the mystery of how these molecular machines literally build themselves.
So, here's our game plan. First, we're going to see how proteins love to team up. Then, we'll uncover the surprisingly beautiful and, well, mathematical rules they follow to put themselves together. We'll also look at what happens when these structures collapse, which is pretty dramatic. That leads us right into the great folding mystery. And finally, we'll explore how science is starting to crack the code on how they find their one perfect shape out of a zillion possibilities.
All right, let's dive in. First up, this idea that a protein is rarely just one long, lonely chain of stuff. Nope, they are super social molecules. They form these complex teams, and that's a concept known as quaternary structure. A perfect example is hemoglobin, the protein that's carrying oxygen through your blood right now. It looks like one big complicated machine, right? But it's actually a team of 4 separate parts, or what we call subunits, all fitting together perfectly to get the job done. And here you can see it visualized in a couple of different ways, as those flowing ribbons, and then as a more solid, space-filling model. So this precise arrangement, this assembly of multiple protein chains into one bigger functional thing, that is what scientists call quaternary structure. You can think of it as the highest level of protein organization, and without it, crucial molecules like our friend hemoglobin just wouldn't work. Now, here's where it gets really, really cool. These teams don't just form in some random clump. They are built with unbelievable precision, and they often follow these strict, elegant mathematical rules of symmetry. The most basic rule they follow is called cyclic symmetry. The easiest way to picture this? Think of a pizza or a pie. Each slice is a protein subunit, and they're all arranged symmetrically around a single point right in the middle. You can see it right here. You could have 2 subunits forming what's called a C2 symmetry, or maybe 3 subunits in a C3 arrangement. The principle is exactly the same. It's just an incredibly efficient way for nature to build a stable structure using a bunch of identical parts. But, nature doesn't just stop at the basics. Things can get way more complex and, frankly, more beautiful with something called dihedral symmetry. This is like taking that pie we just talked about and then adding another axis of rotation at a right angle. You're essentially creating a top half and a bottom half that are mirror images. This trick lets nature build even more intricate things, like a 4-part D2 symmetry or even an 8-part D4 symmetry. By stacking these symmetric rings, you can build bigger, more stable molecular machines. It's brilliant. And for the absolute ultimate in efficiency and strength, nature sometimes pulls out icosahedral symmetry. Just think of a 20-sided die or a classic soccer ball. This shape is amazing because it gives you the biggest possible inside space using the smallest possible outside surface. And guess who uses this incredible geometric trick all the time? Viruses. This right here is the poliovirus. Its outer shell, or its capsid, is a perfect icosahedron, built from 60 identical copies of its protein parts. This creates this insanely strong, self-assembling little container to protect all of its precious genetic cargo inside. It's like a molecular fortress. And this isn't the only play in the viral playbook. While the sphere-shaped viruses love their soccer balls, the rod-shaped viruses, like the tobacco mosaic virus, use a totally different but just as elegant principle: helical symmetry. They build a protective tube by arranging all their parts in a spiral, like a staircase. So we have this incredible, mathematically perfect order. But what happens when that precision is lost? Well, it turns out these beautiful structures are surprisingly fragile. And when they break, they don't just bend, they collapse catastrophically. This whole process of a total structural collapse is called denaturation. It's when a protein loses that specific 3D shape it needs to work, and when it loses the shape, it loses its function. It's basically the molecular equivalent of a complex machine just breaking down into a useless pile of parts. And what's wild is how this collapse happens. Look at this graph. A protein can be perfectly stable one moment, but if you just nudge the temperature up a tiny bit, it doesn't just get weaker, it unravels, abruptly and completely. It suggests this is a cooperative process, where if one little part fails, it triggers a total structural meltdown. It's like a house of cards. This crazy fragility brings up a massive, monumental question. If these perfect structures can fall apart so suddenly, how in the world do they manage to fold themselves so perfectly into that one correct shape to begin with? Well, a Nobel Prize-winning experiment by a scientist named Christian Anfinsen gave us our first huge clue. He took an enzyme, ribonuclease, and just completely unfolded it with some chemicals, totally destroying its ability to work. But then, and this is the amazing part, when he removed those chemicals, the protein just snapped right back into its perfect original active shape, all by itself. The conclusion was stunning. All the information needed for that final 3D structure has to be right there, encoded in the simple, linear sequence of its amino acids. Okay, but that answer just created an even bigger, crazier problem, a paradox first pointed out by Cyrus Leventhal. He asked, all right, if the protein has to find its one correct shape by just randomly trying out every single possible fold, how long would that actually take? The answer is, well, it's just mind-bending. For even a small, simple protein, it would take an estimated 10 to the power of 77 years. That is a number so big it's meaningless. It's billions and billions and billions of times longer than the entire age of the universe. And yet, real proteins fold in seconds, or even faster. So clearly, a random search is out. It's an impossible timeline. There have to be shortcuts. There must be pathways or a set of rules that guide the protein to its final destination without trying everything. The big question is, what are those rules? So how do they pull it off? Well, scientists have a couple of big ideas. One is that they build in stages. Small local bits, like little corkscrew helices, form first, and then those pieces kind of find each other and click together. Another idea is the hydrophobic collapse. Basically, parts of the protein hate water, so the whole thing really quickly scrunches up to hide those parts on the inside. This forms a compact little blob that then fine-tunes itself into the final shape. Honestly, the truth is probably a mix of both. I think the best way to visualize this is the free energy funnel concept. Picture the unfolded protein at the top of this wide funnel. It has tons of possible shapes it could be in. That's high energy, lots of chaos. As it starts to fold, it's guided down the walls of the funnel, which narrows the number of possibilities and lowers its energy. It avoids getting stuck in little traps along the way until it reaches that one single, super stable shape at the very bottom. And look, this is not just some fascinating academic puzzle. This has real, actual life-or-death consequences. When this folding process goes wrong, even just a little bit, the results can be devastating. A whole range of horrible human diseases, including cystic fibrosis, are ultimately caused by a single kind of protein that just can't find its correct shape, and therefore can't do its critical job. And that leaves us with a final, kind of provocative thought to chew on. We've learned that a simple, one-dimensional string of amino acids contains the complete blueprint for a complex, self-assembling, three-dimensional machine. It really makes you wonder, what other profound secrets about life itself are hiding in plain sight, encoded in these biological sequences, just waiting for us to finally figure out how to read them